Hi, good morning everyone. Thank you for joining us in this sharing sessions on next generation sequencing and its applications. I'm Jackie from NextGene Scientific. Feel free to drop us any questions in the chat box and I will try my best to answer them. Without further ado, let's begin. First of all, let us begin with the evolutions of sequencing technologies. We have 3 generations of sequencing technologies up to date. The first generation sequencing is very common and popular and is also known as Sanger sequencing. Some even call it single pass sequencing. Sanger sequencing utilizes capillary electrophoresis to determine each and every bases in a DNA, and it has a reef length of 500 to 1000 base pair. This is the first generation sequencer, the ABI 3730XL, and it can run up to 384 samples in a run. Then we have the second generations and third generation sequencing. We refer both of them as next generation sequencing. Next generation sequencing is high throughput. Instead of running 384 samples or reads at a time, NGS is running billions of reads in a run at a time. As a result, we are getting much more information compared to the first generation sequencing. For second generations, we have a few major companies, for example, Illumina, MGI, Ion Torrent from Thermo Fisher. And for third generation sequencing, we have Oxford Nanopore and PacBio. The major difference between second and third generation sequencing, it would be the reef length. Due to the limitations of technology, second generation sequencer reef length is within the range of 50 to 500 base pair, whereas for third generation sequencer, the reef length on average can reach up to 10 of thousands base pair in one read. For Oxford Nanopore, the longest reef length achieved is 4 megabases.
So, what is NGS? As mentioned earlier, NGS is a high throughput and massively parallel sequencing technology. It is used to determine the order of nucleotides in entire genomes or targeted regions of DNA or RNA. NGS is also scalable and cost-effective due to large amount of data produced. Researchers can apply NGS in many different types of scenarios according to their experiment needs. Despite all the advantage of NGS, Sanger sequencing still remain, or the first generation sequencing still remain the gold standard if you are targeting a few gene or amplicons. However, if you have multiple targets to study, NGS would be a better choice. Then we have a comparison between short read sequencing and long read sequencing. In order to get long reads for sequencing, you must use high molecular weight HMW extraction kit to extract your samples. This is because normal extraction kit with column will fragmentize your DNA, and you cannot get long fragment for long read sequencing. As mentioned earlier before, also the read length for long read sequencing is up to 4 MB for now, but maximum 500 base pair for short read sequencing only. With a long read sequencing data, it is also easier for bioinformatic analysis. This is because some region of the genome are actually challenging for short read sequencing data. For example, high GC region repeats. With the long read sequencing data, these regions can be overcome easily. For sure, short read sequencing also has its own advantages, which is high accuracy and high sequencing depth. It is also cheaper to perform short read sequencing at the moment. Long read sequencing also has a few perks. For example, Oxford Nanopore platform can perform adaptive sampling for targeted sequencing, direct RNA sequencing, sequence modified and methylated bases without the needs of bisulfite conversions. And this is the common workflow for NGS. First, we have to prepare our samples. For example, DNA extractions or RNA extractions. reverse transcriptions, PCR. Then we have to add a barcode to our adapter, or we have to add a barcode or adapter to each of our samples. This is because in NGS, every run can produce large amount of data. So multiplexing of samples is a very common practice. The barcode helps to identify the reads are from which samples. Then we will sequence the samples with one of the NGS platforms mentioned earlier, Illumina, MGI, Ion Torrent, Sentinel-Paul PacBio. After that, we will get the raw data, and then we will need to perform bioinformatic analysis to turn all those sequences into meaningful data. Let us move into applications of NGS. Here are some of the common NGS applications. For example, metagenomics, whole genome sequencing (WGS), whole exome sequencing (WES), transcriptomic (RNA sequencing), chip sequencing, single cell sequencing, and more. We will go into details of every application listed here, and also share some case studies for the applications here. First, let's talk about metagenomic. Metagenomic is the study of genetic material recovered directly from environmental sample, and it does not require a pure culture. You can extract the genetic information or genetic material from soil, water, feces, gut, oral, and etc., and directly sequence whatever is inside the samples. It saves you from a lot of culturing work, and in conventional way, you might also miss some hard to culture species. There are three types of metagenomics: amplicon sequencing, shotgun metagenomic, metatranscriptomic. In amplicon sequencing, we amplify the 16S or 18S or ITS region to determine the microbial biodiversity in a sample. But because we are targeting these amplicons only, the information we get are also very limited. This is what you can expect from the data. Who is living inside the sample? In this case, different bacteria. It means bacteria. But for shotgun metagenomics, we can get answers for who is living in the samples and what are in the samples. This is because we randomly sequence all the DNA present in the samples. Unlike the previous two types, metatranscriptomic is randomly sequencing the RNA found within an environmental sample. As we all know, RNA still will translate into enzymes or proteins, and this gives us an idea on how they do it. For example, you found a soil sample which can degrade plastic. A big finding, right? But what enzyme or genes is responsible for decomposing the plastic? So this is where metatranscriptomic can help. This is an agriculture case study where they study the effect of soil death on the structure of microbial community in agriculture soil in Iowa, USA. Researchers noticed that microbial biodiversity changes with the depth of the soil. When they dig deeper, they found this proteobacteria is increasing in number. On the other hand, metagenomic can also be applied to healthcare research too. In these studies, researchers are able to get high coverage whole dengue and chikungunya virus genome direct from clinical samples. This is very important, especially when there is an outbreak. We must know our enemy well in order to fight it. The metagenomic approach can detect unexpected causes of disease in the midst of others' outbreak, as well as co-infections. It is ideally suited for the investigation of viral species with high level of genetic diversity, which are difficult to access using targeted methods. In these studies, researchers are using Oxford nanopore sequencing to detect the correct predominant virus in all cases, even at low concentrations, and was able to detect cases of co-infections. Then we have another metagenomic case studies for healthcare. This is a paper published by UITM researcher, where researchers study the gut microbiota in pregnant Malaysian women. They collect fecal samples during the first trimesters and third trimester during pregnancy, and they perform 16S amplifications. They perform 16S amplicon sequencing to determine the gut microbiota in pregnant women. They noticed several key bacteria from these three classes has potential metabolic links with gestational diabetes mellitus (GDM) during pregnancy. GDM is a type of diabetes happen only on pregnant women due to the changes of hormones. The next part, we move into whole genome sequencing. Whole genome sequencing is used to determine the entirety or nearly the entirety of the DNA sequence of an organism genome at a single time. Large amount of information generated make WGS a very powerful tool in genomic research. For example, characterizing mutation, identifying inherited disease, tracking disease outbreak, personalized medicines, selective breeding, and etc. One of the largest WGS project would be the Human Genome Project. Initiated in the 1990s and completed in April 2003, for the first time, we are able to read the genetic blueprint for building a human being. Although this first draft of human genomes published in 2003 was a landmark achievement, but it was also full of holes due to the technological barriers. The rising of high accuracy long read sequencing, third generation sequencing, has finally removed this technological barrier. Telomere to Telomere Consortium combines PacBio Hi-Fi and Oxford Nanopore Ultra Long Read Sequencing to address the remaining 8% of the genome and present us a complete 3.05 billion bases pair sequence of a human genome in 2021. They also annotated few thousands of genes which are missing during the first draft human genome. Although CHM13 represents a complete human haplotype, but it does not capture the full diversity of human genetic variations. This is because CHM13 is a Caucasian female cell line, hence the missing of Y chromosome, and it does not represent Asians or African genetic variations. To address this bias, the Human Pan-Genome Reference Consortium has joined with the T2T Consortium to build a collection of high-quality reference haplotypes from a diverse set of samples. And this project is still ongoing. In this whole genome sequencing case study, researcher from National Health Service UK is able to detect patterns in the cancer's DNA, also known as mutational signatures. WGS data give us an idea in how the cancer may have developed, how it will behave, and what treatment options would work best for the patients. We have another case studies in WGS. In this WGS case study, researchers are studying the TB bacteria. They use Oxford Nanopore to sequence the genome of an extensively drug-resistant tuberculosis bacteria, and they are able to identify the mutation which are responsible for the drug-resistant characteristics. Unlike the previous two case study, this case study is on agriculture. Oil palm is one of the crop which is extremely important to Malaysia economy, as we export a lot of palm oil. Therefore, MPOB initiated the Malaysians oil palm genome program to produce a draft genome of the oil palm to produce a draft sequence of the oil palm genome. 3 oil palm genomes were assembled and annotated by this program. The objective of the programs are to obtain a gold standard reference of the oil palm genome, to identify markers and genes or promoter of interest associated with important agronomic traits. To use the genome informations to develop disease-resistant palm with enhanced productivity, to develop value-added products of oil palm. This is another case study with WGS in animals. China researchers successfully de novo assembled the genome of Trachidermus fasciatus, also known as the rough-skinned scorpion fish. Due to overfishing and environmental disruption, the population of rough-skinned scorpion fish is currently under threat in China and listed as the Class 2 protected animal. It is very important to have a reference genome to study the population genetics, domestic farming, and genetic resources protections. China researchers integrate nanopore long-read sequencing, Illumina short-read sequencing, and high-C methods to thoroughly de novo assemble the genome of Trachidermus fasciatus. This studies is very important because it provides the first whole genome reference for the species of Trachidermus fasciatus and might be important for future studies for the protection and domesticate culturing of Trachidermus fasciatus. Then we move into whole exome sequencing. Instead of sequence the whole genome, we can sequence all of the protein coding regions of genes in genome, also known as the exome. Humans have about 180,000 exons, constituting about 1% of the human genome, or approximately 30 million base pairs. WES is used to identify all the genetic variants that alter protein sequences. Exome sequencing can also be used to diagnose the genetic cause of disease in a patient. For example, Alzheimer's disease. First, the exome is being captured, then enriched, and subsequently sequenced. In this WES case study, researchers from IMR studies the clinical utility of WES for inborn error of immunity. WGS enables the detection of structural and deep intronic variants. However, WGS data is less practical for diagnostic testing due to its massive data output and high cost. Compared to WGS, data output from the WES is more manageable as it sequences approximately only 2% of the genome that harbors approximately 85% of disease-causing mutations. These studies show that WES enhance the capacity to diagnose inborn error immunity, also allowing more patients to receive appropriate therapy and disease management. I would like to share one of my personal experience. I'm not so sure if this happened to our audience before, but when I'm on my social media, I always saw this advertisement for personalized genetic testing pop-ups. In fact, many of these testing service providers are utilizing WES to study our healthcare profile and give you a report. With our genetic information, service provider then predict our cancer risk exposure, health risk or disease risk, drug responses, allergic, and even provide advice and even provide advice for our dietary to maintain a healthy lifestyle. In the next part, we have transcriptomic sequencing, and also known as the RNA sequencing. RNA sequencing reveals the presence and quantity of RNA in a biological samples at a given moment. RNA sequencing can tell us the difference in gene expressions in different groups or treatments. RNA sequencing is also used to study alternative gene splice transcripts, post-transcriptional modifications, gene fusions, mutation, or SNPs. But please not to confuse with total RNA sequencing. RNA sequencing above is referring to messenger RNA sequencing only, mRNA sequencing. Researcher likes to study mRNA because mRNA will be translated into proteins or enzyme and eventually change the phenotypes. Of course, we can also study others RNA with NGS. For example, microRNA sequencing, long non-coding RNA sequencing, and many on. Or we can study total RNA sequencing with NGS, which combines a few sets of data from different types of RNA sequencing. This is a case study by UPM researcher. They utilized RNA sequencing data to study oil palm defense mechanism in response to a fungal invasions. They found 7 genes in oil palm that often upregulated during an fungal invasions, and these 7 genes might potentially to be developed as the biomarker for the early detections of basal stem root disease in oil palm. Then we have chip sequencing. Chip sequencing is also known as the chromatin immunoprecipitation sequencing. It is used to identify genome-wide DNA binding site for transcription factors and other proteins. Chip sequencing is used to study the interactions between protein and nucleic acids. Protein bound to DNA are fixed and isolated with chromatin. Then subsequently, we digest the protein, and subsequently the DNA. Then subsequently we sequence the DNA that are initially bound to an proteins. Chip sequencing reveals insights into gene regulations events that play a role in various diseases and biological pathways, such as development and cancer progressions. The next applications, we have single-cell sequencing. Single-cell sequencing is to study the genomes of transcriptome of individual cell, providing a high-resolution view of cell-to-cell variation. Individual cells can exhibit substantial differences in gene expression, protein levels, and phenotypic output with important functional consequences. We isolate each cell and measure its specific contribution to the whole cell populations. For instance, single-cell RNA sequencing has been used to investigate rare antigen-specific T or B cell, study the origins and development of chemo-resistant tumor subpopulations, discover previously unknown gene function in plant tissue, study tumor progression mechanism, and many more. In recent years, single-cell sequencing has gained popularity to study cancer cells and diseases, and we share a case studies on comprehensive single-cell analysis on influenza A virus infections. Researchers infect the human airway epithelial cell with wild-type and mutant influenza A. Then single-cell RNA sequencing is performed to study the immune response of different cell types. Influenza A induce dynamic and cell type specific transcriptional changes that occur on a single cell level at the nature site of infections. This study embodies the first step in generating a comprehensive overview of the complex virus-host interaction within the heterozygous cellular compositions of human respiratory epithelium. Due to the time constraint, we are unable to share the full potential of NGS. Some of its other applications include cancer cell panel studies, non-invasive prenatal testing (NIPT), pre-implantation genetic screening (PGS), and also precision medicine study. Precision medicine study is to tailor chemotherapy drugs for patients that does not respond to common cancer drugs. In summary, NGS is a high throughput method to determine the sequence of DNA or RNA. Short-read sequencing has higher accuracy, long-read sequencing simplifies the bioinformatic analysis part. NGS has various applications in genomic studies, and designing an NGS experiment can be a complicating process. User must choose the right application, the right sequencing platform, the right coverage or sequencing depth, the right data output, and bioinformatic analysis. If you are in doubt, feel free to contact our sales team. We are more than happy to share our experience with you and guide you through your NGS journey. And this is the end of my sharing sessions. Thank you so much for joining us and hope you have learned something today.
Thank you, Jackie. Is there anyone got any questions? So we got one question from Dalila Zarifa. So the question is, so if for COVID WGS, will it be short or long read?
Answering Dr. Zarifa questions. So for COVID WGS, it can be the short read sequencing or also the long read sequencing, depending on what platform you are using. If let's say you are using Illumina platform, then it will be short read sequencing. But if let's say you are using an Oxford Nanopore platform or even PacBio platform, then you are doing long read sequencing data.
So the next question is from Muhammad Naim. The question is, what is the best tool to use for the library?
To reply these questions, we must know what the applications you are doing, and then also because library preparations kit, many companies or many brands also offer the same thing, but it still depends on your application. So if let's say you would like to know more about on this, you can contact our sales team, and then we can guide you through.
We have the third question from Karen. What is a draft genome sequence? Why can't it be considered as complete?
Okay, so for draft genome sequence, actually we means you assemble the data from a short read sequencing, and then because of some of the regions is hard to sequence, or because due to the limitation of technology, we cannot sequence the region. So there is a hole or patches at the genome. If let's say we were to consider it as a complete, we have to use long read sequencing data to compensate or to match all the holes in between, because this region, this hole or context, actually maybe they have some information which are important, so we cannot consider a draft genome sequence a complete genome.
Okay, so we have another question from Liana. For RNA-seq, can we only sequence certain target genes instead of the whole genome?
For RNA-seq, actually you are sequencing the all of the genes, all of the mRNA genes you extracted. So is how to say, because it's a snapshot. The RNA sequence at the moment is actually a snapshot of what are the genes are being expressed at the moment. And if let's say you are only targeting a few genes, then maybe qPCR would do better. But this is RNA sequencing is mainly used to study difference of different gene expressions profile. And of course, you can target. You can fish out the few genes that you are interested from the RNA sequencing you did. Also can.
Thank you, Jackie. Is there any other questions? So we have another questions from Muhammad Ramzuddin. Dear speaker, to go for complete genome sequencing, which platform that will be suggesting the most? Is it either Illumina, such as MySeq, HiSeq, NovaSeq, or Nanopore, or PacBio? What are their brief advantages and limitation? Thank you.
Okay, so for a complete genome sequencing, I'm not sure here you are referring to a bacteria or eukaryotes. If a bacteria, you can go for Illumina or also Oxford Nanopore, or PacBio because Oxford Nanopore and PacBio will have an advantage as it can sequence the whole bacteria genome maybe in one contig. In one long read, you can sequence the whole bacteria genome. So you will get a genome without gap. But for, if let's say, eukaryote complete genome sequencing, or you're referring to bacteria, okay. So, but also share lah. If let's say eukaryotes, actually you can do what the if let's say you need a very complete genome map for eukaryotes, you can do what we just now the case study I showed, the China researcher to do combine the Illumina data, PacBio data, Oxford Nanopore data, and also HiSeq data to get the complete whole genome of a eukaryote. But of course, this will be a very big and costly project, I would say.
Okay, the next question is from Priya. It's regarding the metagenomics. If I were to compare the bacterial population using the 16S sequencing and shotgun metagenomic, would it be a vast difference?
Yes, it's a very vast difference. For 16S, because we are only targeting the 16S, V3 and V4 region, so the information you get is very limited. You can only get what bacteria is inside the samples, because as I mentioned before, who is living in the samples. So you can get the microbial biodiversity of the bacteria, so you can get a pie chart or bar chart to see what are the majority or minority bacteria in your sample. But for shotgun metagenomic, you can get the microbial biodiversity and also what genes is inside the samples. So for example, if let's say you are working on antibiotic gene resistant or antibiotic gene discovery, or maybe you are looking for some of the gene is inside the samples, then shotgun metagenomic will help. But 16S sequencing will only let you know who is inside the samples. And go back to the previous questions. So what are the brief advantages and limitations? The advantages for Illumina or short read sequencing would be the high accuracy and is cheaper currently. But for Oxford Nanopore and PacBio, although it is more expensive, but then they can get a complete genome map, because a long read can actually covers all the regions.
Thank you, Jackie, for the comprehensive answer. Is there any more questions from the audience?
So thank you, everyone.
So actually, we got a special promotion for our NGS package now. For anyone who subscribe for the NGS service for more than 10K, you will have a free set of accurate micropipette sets and a pipette stand, which worth 2,005 with the term and condition applied. So anyone got any inquiry, feel free to drop us an email or find our sales representatives in your territory. And for our upcoming event, we are holding a Malaysia Next Generation Sequencing Symposium. So the title will be the Unraveling the Genome and the Future. The time for this event will be 14 to 15 September, and the venue will to be confirmed yet. Anyone who interested or want to get to know some any information, you may drop us any questions in our feedback form later. So thank you everyone for attending this event. Wish you a good day ahead. Thank you and see you again.
Thank you.